Hello, and welcome to today's webinar, Optimizing Movement Disorder Clinical Trials, a focus on Parkinson's disease. I'm your host, James Crosspayne with Clinical Leader. I'm joined by two experts from Dr. Vince Clinical Research. They are Pete Schmidt, Chief Scientific Officer, and Amber Anderson, Senior Project Manager. And we're joined by Polly Dawkins, Executive Director of the Davis Phinney Foundation, the moderator for today's fireside discussion. I'd like to thank you, the audience, for attending today's event. Take a moment to get comfortable with the tools on your screen. You'll find your presenter's contact information, as well as a question box. Please share your thoughts with us during the presentation. We will answer as many questions as possible, and any we miss, I'll pass along for follow-up after the event. This question box can connect you with me if you run into any trouble, but please try refreshing your browser first, as it will fix most common issues. Our presenters have also asked a few questions of you that are located on the page. Please take a moment to share your experiences with us. Finally, today's event will be available on demand. You'll receive an email with a shareable link shortly after we conclude. Now, without further ado, let's begin today's discussion. Welcome, Polly.
Thank you, James, and thank you to Dr. Vince Clinical Research for having me here to moderate this conversation. I'm excited to have this conversation with Dr. Schmidt and Amber Anderson. First, I'd like to ask Dr. Schmidt, may I call you Pete?
Of course.
Super. Pete, you've been in Parkinson's trials for a long time. Can you give us a little background of what's going on in Parkinson's disease clinical research right now?
Absolutely. So this is a very exciting time in movement disorders research. We've reached what I think of as sort of the third revolution in our understanding of Parkinson's disease. The first revolution was the discovery of the neurotransmitter dopamine and the dramatic decline that we see in patients with Parkinson's disease. This was built on a foundational discovery by Arvid Carlsson, work for which he won the Nobel Prize. But it was just as important the work done by Ole Hornakewicz, who figured out clinically how levodopa could be administered to treat Parkinson's disease and got us started on the path to the first effective treatment for the disease. Almost everything we do today clinically for Parkinson's patients builds on this insight. We've since followed dopamine signaling through the deep structures of the brain to understand how the direct and indirect pathways are involved in dopaminergic networks, insights that led to Malin DeLong's breakthrough with deep brain stimulation. The second revolution came in the discovery of alpha-synuclein. Lewy bodies have been associated with Parkinson's disease since the First World War, but geneticist Michael Palomopoulos found an Italian family with the mutation in the SNCA gene. And a team including the late Penn neuropathologist John Trojanowski figured out that the principal component of the Lewy body was the protein coded by that gene, alpha-synuclein. This was a very interesting insight because the SNCA gene is not a major cause of Parkinson's disease. So it was just luck that we found that gene first. With a push by Stan Prusner, all of us who focused on neurodegenerative diseases that have a characteristic protein, which is most of them, turned their attention onto the protein life cycle. Well, the pathology is usually linked to off-target impacts of protein accumulation. Cullen and Crank each identified mechanisms explaining Parkinson's disease link to risk genes related to protein turnover. We've all seen other factors linked to synthesis, folding, and function. And people hear a lot about misfolded alpha-synuclein, but misfolded alpha-synuclein is only one part of this puzzle. The third revolution has been uncovering potential treatments for Parkinson's in our new understanding of how the brain's regulatory systems manage brain health. What you see here is the latest insight, the glymphatic system, which is a convective flow that clears away waste products in the extracellular space and sweeps them into the CSF. New insight into neuroinflammation in the brain's structural matrix are informing other potential druggable targets. We have so many potential targets for interventions and so much new insight into these pathogenic and pathological mechanisms. There really never before been so many different trials that could change the patient experience of the disease and introduce new mechanisms and new targets into our armament for how we fight it.
A lot of what you're talking about, mechanistic trials, recruit people with Parkinson's who are newly diagnosed. Isn't that correct?
So most of these mechanisms, because they focus on how the disease starts, are most effectively tested in patients who are newly diagnosed with Parkinson's, because as a neurodegenerative disease, the attack surface of pathogenic mechanisms gets reduced over time. So I usually tell, if you've got a drug that's targeting the pathogenic mechanism, I usually tell sponsors they should recruit patients with less than 5 years since diagnosis.
That makes sense. So let's talk a little bit about symptomatic trials, because they're focused on people affected by Parkinson's right now. And Amber, you've worked on quite a few symptomatic trials, haven't you?
Yes, I have. And based on the symptomatic trials I've worked, there's some things you want to keep in mind. First off, you want to think beyond just today's patients. Your recruitment planning in these trials starts well before you start enrolling. It's your feasibility. You want to really look at the current ongoing population, but also future. Look at a site. They may be seeing appropriate patients today, but then 5-6 months down the road, if enrollment starts, they may no longer be available or eligible. So we want to look at today and in the future patients. Another consideration is the patient experience. These symptomatic trials can be shorter than other trials. Your participants may notice improvements in their symptoms relatively quickly. And when someone feels like this treatment's making a difference, they may naturally be more engaged throughout the study, and it in turn is better for retention overall. One thing though we need to be careful on is not every patient will respond in the same way or have the same experience. And while we want to be excited about these benefits of the treatment, we don't want to create unrealistic expectations for some of the patients. Just remember, operationally stay consistent, follow your protocol, and you should be good there.
Okay, that makes sense. So Pete, can you tell us a little bit about what symptoms are being targeted in these symptomatic trials?
So a major focus has been improving levodopa response. Patients who are well managed under levodopa still may experience some side effects of medication, like unexpected motor fluctuations, and also dyskinesia. And there have been a number of efforts to try and improve good on time without dyskinesia for patients. But we also see a lot of drugs that will target non-motor aspects of the disease, like depression, anxiety, sleep, psychosis. These things are very important for patients. I was recently talking with a sponsor who's looking at psychosis, and they were talking about the impact of psychosis on the caregiver, the challenges of working with the anxiety that can come with the disease, the hallucinations or delusions that can come with advancing Parkinson's disease, and the impact they can have on relationships. So we're very enthusiastic about the opportunities that there are to provide better management for these features of the disease as well. For many patients, a little dyskinesia or some on-off fluctuations can be manageable, but poor sleep, depression, anxiety can make life very difficult. And so better treatments for those would make a huge benefit for the people living with the disease today.
Yeah, that's what we hear in the community as well, Pete. So let's shift our focus a little bit to disease-modifying trials. How are they different from symptomatic trials?
So first of all, they take they can take longer. Parkinson's disease progresses very slowly, and many times a patient can be newly diagnosed and have some upset, some depression from the fact that they've been diagnosed with a scary brain and neurodegenerative disorder. And then they may, over time, realize that this is a manageable condition and that they can live a good life with the disease. And so you may see some impacts of the disease kind of get less burdensome in the early experience of the disease. So when we look at a disease-modifying trial, it's very important that we think about how are we going to measure progression, which is both slow and sometimes will change. There's a dramatic change in patient status when they start taking medication. They get immediately much, much better from the perspective of the neurologist. Their motorically, they improve dramatically. They get a lot of benefits from the administration of levodopa. And so we'll see that those patients get a lot better. And if you look at with early disease, because that's what we usually recruit for these trials, you will see that the patients the patients who were recruited into the PPMI study, which is a very comprehensive study of early disease sponsored by the Michael J. Fox Foundation, they found that many of their patients in the motor skills showed improvement from baseline to 2 years out. About 40% had a better UPDRS score 2 years after diagnosis than they did at baseline. 2 years after the baseline study. And that result was, it makes it very hard to see this progression in a disease-modifying trial, especially because very few of these trials have the potential to reverse the disease. Most of the time we're looking at slowing or stopping. There are some trials that we're looking at where we expect there'll be a symptomatic benefit, where some aspect of the disease will improve based on the symptomatic treatment, based on the disease-modifying treatment. And what we look for there is some of these more subtle features like sleep or maybe anxiety could benefit from a disease-modifying treatment, and then we may be able to find a result faster.
Yeah, Amber, do you have any thoughts on disease-modifying trials and in keeping people engaged in these trials?
Yeah, as Pete mentioned, these trials do tend to take longer. Sometimes they have more restrictive eligibility criteria, and this can make recruiting and retention of disease-modifying trials challenging. Sometimes these items, can place greater burden on the sites and the participants, especially and then retention-wise, with it being disease modifying, the patients might not feel the benefit or see, how it's benefiting them, the treatment is. And so, if they can't feel or see it, sometimes they get discouraged and may not want to continue, the longer duration type of trial. So that adds to your retention challenges.
Yeah. But as Pete said, maybe there are some symptomatic benefits that somebody may experience in a disease-modifying trial.
Yeah. And I would imagine those would go, a lot better retention-wise, if they're seeing and feeling.
But we would be thrilled to have a drug that could slow the progression of Parkinson's disease by 25%. And yet, because there would continue to be progression, the patients may not be aware that they're getting that benefit. And we could show that clinically by showing that our group of patients that were getting the drug were progressing more slowly than the group of patients that weren't. But both sets of patients would be progressing. And this has been the experience in some of the Alzheimer's drugs that have been approved, that have gotten FDA approval. But what really happened with them was that both groups advanced, just the the treatment group, the arm that was receiving the study drug, advanced more slowly.
Got it.
Okay.
I know many of these trials that we're talking about are looking to recruit de novo patients. Amber, can you tell us a bit about recruiting de novo participants in these trials?
Sure. Recruitment really starts with understanding exactly who the protocol is asking us to enroll. For our de novo patients, the eligibility criteria can be pretty specific. So one of the first things you want to understand, like when you're checking feasibility with your potential sites, is, who do you currently have in your practice that might fit this profile? Even patients who aren't immediately eligible when the enrollment's to begin, it'll give us some insight into if the sites routinely see these type of early stage patients that we'll need over the course of the enrollment period. We also must recognize these patients are often eligible for multiple trials, so your recruitment can be competitive. It's very important that your PIs and your site staff understand not just the protocol, but why this your specific trial you may work on is important, and to make it competitive against any other competing studies out there. When your PIs and site staff can confidently explain why the study matters to the participants, they're more apt to feel like, oh, it's meaningful. I really want to participate in this one. Beyond the sites themselves, recruitment's kind of a multi-pronged effort. We often partner with recruitment vendors to reach patients who may not be connected with a study site in particular, but we also work with Parkinson's advocacy organizations to help build awareness and educate the community about different research opportunities. Your strategy also depends on the size of the trial. If it's a larger program, you want a national outreach campaign. Those make more sense because you're trying to reach a broader population. For smaller studies, a much more targeted approach. You want to focus on high performing sites and local patient advertisements and communities. Those can be more effective. But ultimately, successful recruitment of de novo studies, it's not just about finding patients. You want to match the right patients to the right sites, engage with your sites, and then build trust within the Parkinson's community.
Yeah,
so another factor to consider would be what does the referral network look like at the site? Is the site part of a large health network where new patients, when they're identified as potentially having a tremor or movement disorder, get referred to an expert neurologist, the kind of places where we run trials, those sites will be more likely to have de novo patients. Many expert sites will wind up being a tertiary referral site. So the patient gets seen in a primary care setting. In that primary care setting, they may start the medication and only pass that patient on to a neurologist when they feel like the care is getting challenging. In other areas, patients get referred to a neurologist early, which is really considered to be a best practice. In the UK, the expert center guidelines suggest that patients be referred to a expert, a subspecialty movement disorder neurologist early and before initiating anti-Parkinson treatment. And I think that that's a best practice. It would also, in regions where that does happen, it also helps us recruit into de novo patient studies.
Amber, I can imagine once you've recruited people into trials, another challenge is keeping them engaged, retaining them in trials, and tracking them. How do you go about doing that?
So for long-term trials, one of the challenges, like as Pete mentioned, this disease progresses slower, meaning you have extended observation, and you often have to consider, your follow-up and retention strategies. Have those built into your plans and your protocol from the beginning of the trial not just midway or as you go, as things occur. Always remember, retention is just as important as your recruitment. You can recruit all the participants you need for your trial, but if, say 50, 60% of them drop, that doesn't help with what you're trying to achieve with them. So some ways to reduce retention issues are reduce the patient burden, such as allowing for more flexibility or flexible visits. If the participant needs help getting to the site, get them an Uber, a Lyft, bus tickets, whatever you need, depending on the city, but also utilizing technology to help with streamlining visits and assessments, such as like telehealth and virtual visits. You have ePro assessments that can be done on multiple different types of devices, even the participant's own device. That can help you get your assessments done, especially if you have many outpatient visits over the long-term period. Additionally, your sites need to keep the participants engaged by reaching out to them regularly to help establish, a positive relationship with them, checking in on them. It really shows that you truly care. In my experience while in the clinic, these participants appreciate that additional effort that you're putting forth, and it goes a long way to making a difference in their experience in the trial and their engagement and retaining them throughout.
Pete, I can imagine that wearable devices can help in trials, and I know a lot of our community members want to track their symptoms with Apple Watches, other Google devices. Have you had experience with that?
So wearable devices can give us really important information, but there are a bunch of challenges with them. The devices from Apple, they have the movement disorders API, which really does a great job of tracking medication effects. The movement disorders API can identify on time, off time, and dyskinesia very effectively, and probably better than other devices. But often, if we track movement can have a number of different factors that affect a patient's movement. One big one that we often discount is depression. So if a patient isn't motivated to move, the patient movement will show will move less and will show as slower. A psychiatrist friend of mine has told me that we used to think of depression as a psychomotor condition and not just a psychiatric condition. And that motor aspect does show up. And patients can fluctuate between, depression can manifest much more quickly than Parkinson's can advance. And so if we see a change in movement, we need to think about, well, is this patient experiencing a depressive episode? That's why I like to have trials where if there's going to be some sort of quantification of movement, that we include a clinical evaluation as well as at-home ambulatory measurement, because that clinical evaluation will have a person in the room who can understand the mental state of the patient. Is the patient moving more slowly because the patient has low motivation, or is the patient moving more slowly because Parkinson's is restricting their motion? Understanding that state of mind when we're measuring is really critical. And in many ways, that can be a limitation on our ability to gain insight in a device that's sent home with the patient. There are also a lot of logistical challenges that, as the scientist, I don't really think about, but I need to rely on my colleague Amber to think about how do we actually make this stuff happen? How do we keep batteries charged and things like that? Amber, your thoughts?
Yeah. Wearable devices can be incredibly valuable, as you mentioned, but they do introduce some complexities. Some of the common issues we see are, power and data transfers. How long does the battery last? How long does it take to charge? Data sync issues when participants struggle with trying to upload? Does the data automatically go through? What steps do they need to follow? Does it transfer via Wi-Fi, Bluetooth, things like that? You can also experience malfunction, or, sometimes participants need replacements. Also, sometimes the wearables chosen for the trial aren't user-friendly, so participants get frustrated, and in turn, that leads to them not wanting to use them, which brings up some compliance issues. There's some solutions we can do to address this, like providing hands-on training by the site so the participants are comfortable using them, sending home, easy to follow instructions so that if they're at home and, follow the steps, they can upload their data if there are any steps like that for them to take. We want to also, ensure that there's an open communication line for the participant to contact if they're having, technical issues or need a replacement quickly. If your wearable system has like a monitoring or a compliance dashboard, that's awesome. It allows your site coordinators to check daily, and they can intervene early if they notice they're not uploading the data or not using it. They can contact the participant and find out what's going on. There's also many vendors out there that they can handle everything for you. I've worked with one recently, Rune Labs. They're amazing, but pretty much all the site has to do is, the patient enrolls, they register them, and then they just take it from there. They contact the patient. If they need that particular wearable device, they ship it to them. They provide 24/7 support, items like that. So, sometimes finding the right vendor will help ease the burden, on your site, your participants, and the sponsor.
Yeah.
And one more thing to note is that we talk about this as a movement, as an outcome, but in fact, this is really a mechanistic issue. This is really, it does not tell us that the patient feels better. And we need to be very careful about trying to go to FDA and saying that this is a compelling clinical insight. It needs to be paired with an understanding of how the patient experiences the treatment effect, and not just, you can't say, well, their watch told me they were better and FDA will approve. It's got to include a patient experience component.
Yeah, so it sounds like it doesn't reduce, wearable devices don't reduce the operational effort, it sort of shifts it.
Yeah, exactly, Polly.
Let's change gears a little bit here, folks. Let's talk about biomarkers. We hear a lot about biomarkers in the Parkinson's community. Pete, can you shed some light on that topic?
So this is a very important topic, and my colleague, Dr. Sheldon Prescorn, and I just wrote a manuscript that touches on how the FDA is looking at biomarkers now, which is a change from how it's been. For a long time, we've been looking to find a biomarker for Parkinson's progression, and we have not really identified anything substantial. And because Parkinson's progression over a span of 6 months or 12 months tends to be reasonably benign from the perspective of the patient, the FDA and international regulators really want us to say that the patient experience is, that the patient, to record the patient experience and understand how the patient is experiencing the disease in the context of the treatment. What we need to do now with biomarkers is, can we demonstrate the mechanistic effect? If we think that we're reducing neuroinflammation, then we need a neuroinflammatory biomarker. If we think that we're rescuing neurons from cell death, then we need to show benefit on a cell death biomarker. We need to think mechanistically and use biomarkers to explain that the patient benefit is occurring because of the hypothesized treatment effect. We demonstrated in a petri dish or in an animal model that this benefit occurs by changing the cell context, but we then need to validate that it occurs in humans. And that's the pathway to the new FDA goal of approval of drugs with a single phase 3 trial, because they're going to look in that phase 3 trial for a paired clinical and mechanistic benefit. So this idea that we'll come up with a single Parkinson's biomarker is really not in that regulatory pathway. The place where it does matter, where we're validating that the patient has Parkinson's, is going to be for inclusion when we do our diagnosis. So when we do inclusion, we typically will use the GE's DAT scan. We've used, there's been some suggestion that we look at this alpha-synuclein seed amplification assay to validate the diagnosis. Those things are where we'll look for a single diagnostic biomarker, and then we'll have a tailored mechanistic biomarker that's going to result in, that's going to drive that's going to explain that our hypothesis for this, why this worked, is correct, and that the patients really are getting the benefit we think they were getting. But a lot of these things have a lot of logistical complexity in terms of setting rules for sample collection and specimen collection and referral to another clinical site where a procedure may be conducted. There's a skin punch assay that's being used to look at alpha-synuclein, which also involves a little bit of complexity. And I let my colleague Amber handle all of that aspect of the biomarker collection and management.
Yeah, Amber, don't you have to collect CSF to be able to, or aren't biomarkers found in the CSF?
Yeah, CSF contains, the biomarkers. We see that a lot, along with, the skin biopsy, as Pete mentioned. But yeah, for CFS collection, skin biopsy, your biomarkers, it's more than just a procedure. It's a whole workflow. There are some challenges when collecting CSF and other biomarkers. So some things you want to consider are, standardization. You want to standardize your collection, your processing, and your shipping across your sites, and checking out what vendors are capable of. I know for especially CSF, it can be pretty invasive for participants. You want to make sure your sites have experienced personnel. They can, perform the procedure. You want to consider, if they don't do it a whole lot, do they need more training, certification, et cetera? But yeah, for invasiveness, if your site is experienced, it might help a little just by sitting a patient down. They may be on the fence, I don't want to participate in this because, this scares me, or it might be uncomfortable. If your site and PI are experienced in CSF collection, you can sit them down and be like, hey, we're trained, we're certified, we've done this X number of times. It may help put them more at ease and be more willing to at least try. So definitely not an easy one, but it is manageable, and we've done it many, many times. So
great, thank you.
So the other challenge with CSF is that CSF gets its protein load from the glymphatic system that we discussed earlier, and the glymphatic system operates during sleep.
So that's a great segue to our next topic that I wanted to ask you about. You had mentioned earlier some of the symptoms that are troublesome for folks with Parkinson's. The importance of sleep is such a big part of improving quality of life. Tell us a little bit about that. What can we do about it?
So we talk about things in a patient's life that may change their experience of the disease. One of them is exercise, and a second one is sleep. So good sleep quality is when the brain will clear out its proteins and transition them from the intracellular space and into the CSF. And so your biomarker signal will be impacted by sleep quality. And a good treatment may result in a good disease-modifying treatment may improve sleep. And that may be the first thing we see. And that may make the biomarker profile more complex. Sleep is a very complex issue in Parkinson's. For many patients, the earliest symptom is going to be REM behavior disorder, when the patient will act out their dreams. The sleep paralysis that happens during REM sleep is impaired, and the patient will move according to what they're doing in their dream. And this can occur before diagnosis. It can occur after diagnosis. It's a complex sleep issue. The things we could do to improve sleep will be very valuable to the patient community. So a symptomatic treatment that improves sleep could have many positive effects across the disease, across the experience of the disease: reduced fatigue, reduced better quality sleep, but also better quality protein clearance from the brain, and possibly a disease-modifying effect.
So Amber, how do you go about introducing sleep in a clinical trial?
So in most of the trials, I've seen them done where you can run a formal sleep study that can give you valuable information. They can be, complex at times though, and costly. So you want to take that into consideration. I think, most of the time I've seen it addressed via questionnaire on sleep quality or the participant's daytime sleepiness. So that's typically what I see.
So a questionnaire about sleep quality could be very effective. Because of the REM behavior disorder, often it's difficult to get the care partner, the spouse of the patient. They may not have good insight into the patient's sleep quality because they may sleep separately. There are patients who talk about, there are spouses of patients who talk about getting clobbered during REM behavior disorder. So in many cases, we can use a physician global impression of fatigue as a good measure for sleep quality. And I've written a paper about using physician global impression in treatment of REM behavior disorder. And that can be an effective measure because it's probably not going to be your primary outcome. It's probably going to be something that you measure and look at in a secondary or exploratory analysis.
So I know that the MDS-UPDRS only measures sleep with one or two questions, maybe daytime sleepiness and sleep. Do you think that trials are really measuring the right outcomes and the things that are important to people with Parkinson's?
So the challenge is, I think that the MDS-UPDRS is actually quite good for measuring the experience of a patient who's got mid-stage Parkinson's, because it was designed in the mid-2000s when what we were thinking about was treatments that would reverse the disease. We were focusing on symptomatic treatments and the potential to use some sort of cell therapy or gene therapy that could have the potential to reverse the disease. And so it's very granular in that mid-stage patient. The challenge we have is that we're using that measure in de novo patients who will be very low on that scale. They will have, we'll see very low scores and the potential for medications to change a symptom from rarely to never. That's the kind of granularity we see on that scale. And so the MDS-UPDRS is a very challenging one to use in in a study of de novo patients because of that low granularity at the early phases of the disease.
Mm-hmm.
Well, and also you have to consider, the MDS UPDRS Part 3, it requires a lot of clinical experience and consistency with your rate, who's doing the assessments. This can be hard in, longer term trials. I think, consistency is best. One way you could address that is, ensuring your raters are certified by MDS or, Pete, as we've talked about before, using a central rater to do the assessments.
So central rater definitely does help. The challenge with a central rater is going to be a logistical one. Mm-hmm. Can you get the systems in place for that rater to do a video rating early enough that your rater doesn't wind up with a huge backlog of cases? You can't really speed it up. And it takes about a half an hour per subject if everything works perfectly. So it's very challenging. It can be very challenging if you don't get that going and stay on top of the backlog of subjects to be centrally rated.
Agreed.
That gets much more complicated as the trial gets larger. You need to pick a proven system that is going to work to manage those videos and getting them across to your rater. So it's very challenging from a logistical perspective.
I think about the stories you've told me, Pete, about what happened after the Salk polio vaccine was introduced. And for the patient advocacy community, that wasn't terribly great for them. After the vaccine was introduced, there were no new cases of polio, but you still had people who were living with polio for decades afterwards. Isn't there a risk of that now with Parkinson's, for the Parkinson's community?
So this absolutely is a big challenge. It's been in the news because of Mitch McConnell's recent fall and treatment for his fall, and people have talked about his post-polio syndrome. I think that this is less of a challenge because it's unlikely that anytime soon we're going to eliminate Parkinson's disease. Mm. What we're most likely to see is slowing of the disease. And so we will have people live with symptoms, hopefully in a well-managed state for longer. And so we will need to have drugs to manage their symptoms, and there will be value in innovation in symptom management. So I'm very enthusiastic about any trial that I see coming out, whether it's disease modifying or symptomatic. I'd be very enthusiastic about people focusing on antidepressant, antipsychotic, sleep, treatment of sleep. As you know, the medications that are necessary to help a patient to exercise can be so empowering to our community. And it's not just motor effects that can reduce a patient's ability to exercise. It can also be poor sleep. It can also be poor depression, poorly managed depression, and apathy. Mm-hmm. Absolutely. So we need to look at all these things. There are great opportunities to provide a benefit. There are drugs that exist today that have the that were developed targeting a different disease, but have the potential to be more effective in Parkinson's disease, and that's a great opportunity for industry, also potentially for foundations and for NIH to consider.
Yeah, great. All right, I've got a lightning round of questions for the two of you. To start off, Pete, what is one common misconception about Parkinson's disease clinical trials?
So I think one common misconception is that it's all about dopamine. There are lots of other ways that we can be thinking about treatment of Parkinson's disease besides just focusing on the dopamine system.
Amber. what's one operational lesson that every sponsor learns the hard way?
Their protocol design. Well, of course, we want it to be scientifically sound and meet, applicable guidelines and regulatory requirements. It's sometimes they realize, the have to have versus the nice to have. When designing, ensure you're targeting, the right patient population without making your eligibility criteria too restrictive. That'll make your recruitment, very difficult. And I have seen that, happen. Also, additional, tons of exploratory assessments, and that can make excessive burden on the site and the participants. So just, that's one thing they want everything, and then, it doesn't go to plan. So.
Got it. Pete, what's one biomarker you'll be watching over the next 5 years?
So NFL, neurofilament light, is a very interesting biomarker. It's already been recognized by the FDA for approval of drugs for other conditions. And I think that there's great potential for that as an indicator of neuronal cell death in Parkinson's disease. It's a very interesting biomarker, could be in a natural history study as well as in a drug trial.
Be watching for that. Amber, we've talked about recruitment. What are the barriers to getting people with Parkinson's into clinical trials? What's standing in their way?
I think, the biggest barriers are awareness and overall, the burden of participating. Patients may not know the trials are available. They might not meet all the criteria. If they find out they are, they might not meet the criteria, which can be discouraging. Some of them can be concerned with things like, the placebo side effects, travel, as I mentioned earlier, how long the timing commitment is. So like in identifying those barriers early on and making participation as accessible and manageable as possible will help us address some of those barriers.
And Polly, if I could add,
Mm-hmm.
One of the challenges I think we have is that people think they know what is the trial they want to be in, and they hold out for that.
Yep.
The Parkinson's disease is an incredibly complex disease. I've been studying it for years, and I don't know where the next breakthrough is going to come from. It's very challenging to have any idea which mechanism is most promising. If you have Parkinson's disease and your neurologist is running a trial, volunteer for it. Mm-hmm. If it doesn't work out, you could volunteer for another one later. But anything we can do to get definitive answers to whether a treatment works or doesn't work is going to help us advance the field and generate enthusiasm from everybody involved in making sure that Parkinson's disease is an area for innovation. Fully enrolled trials are encouraging to funders. They're encouraging to entrepreneurs who want to start a new company. They're encouraging to big companies thinking about what areas they can promote development in. So the biggest benefit that we can do is not wait, but enroll in trials. If your trial fully enrolls but doesn't succeed in changing the course of the disease, it'll make people have more faith that the next trial will also fully enroll, and maybe that's the one that is going to change our the clinical experience of Parkinson's.
Yeah, and to add from the advocacy perspective, the patient community perspective, we hear people talk about, oh, I get more time with my neurologist if I enroll. I get to see my neurologist many more times a year than I would otherwise. And that's really appealing to individuals. Mm-hmm. We also hear that when there's clear, honest communication about what's expected, what the outcomes are, whether you might be on a placebo, and then after the trial, communication about what happened is really important to individuals who are looking to be a part of the trial. All right. right, one last question. Pete, what's giving you the most hope or optimism about the future of Parkinson's disease therapeutics?
So I come from bench science, and what's so exciting to me is our growing insight into the mechanics of how the brain works with Parkinson's, on the systems affected and the circuits and how they change.
Great. I want to ask each one of you to give our audience a key takeaway for today. Amber, may I start with you? What would be your one takeaway that you'd like our audience to walk away with?
My one takeaway, when designing your trial, please try, wherever possible, keep your patient at the center. While the scientific aspects are very important, you also need to have the participants to give you that data. So try to keep in mind, we're people, and they're waiting for these therapies. They're eager for this treatment, to help them.
Pete, do you have a takeaway for the audience that you'd like to communicate?
So what I'd like to see is, I would love to see people, when they come up with a novel drug that might affect one aspect of Parkinson's disease, that they think about running their trials in Parkinson's. Parkinson's depression, if you can treat Parkinson's depression, then you can treat any refractory depression. Parkinson's depression is a super challenging form of depression, and a benefit to the Parkinson's community for apathy or depression will be one that can expand outward beyond. But a trial run in a Parkinson's clinic, you're working with a great group of patients, a great group of physicians, and great clinical teams. So I'd love to see people who not just think about Parkinson's as an opportunity for motoric trials, but for non-motor trials as well. Can you do some, if you've got a great idea for how you might treat psychosis, if you have a great idea for how you might treat sleep, if you have a great idea for how you might treat apathy, think about running those trials in Parkinson's disease. It's going to be a community that is very supportive of that trial, would be enthusiastic to enroll, and you'll work with great teams and great people.
Thank you all for the excellent discussion. Before the Q&A begins, I just want to remind our audience that you can connect directly with our presenters. Their information is located on the side of your screen. We see a lot of great questions coming in. Thank you so much for that. Please keep submitting those via the Q&A box below me. We're going to address as many as we can, and the rest I'll pass along to our presenters and the Dr. Vince clinical research team to follow up offline. Our first question is asking, what about stem cells and gene therapy?
So we talk about stem cells and gene therapy together because a lot of the really promising things in gene therapy are going to be done to cells outside of the body, and then the cells will be reintroduced. Stem cells and gene therapy are a very promising area. We see a lot of people developing drugs, and we look forward to seeing how they fare in trials. There's been a lot of experience in the past, and there's been a lot of interesting results that have informed our understanding. One big issue with cell therapy has been that academic labs have struggled with cell quality, and we've now seen companies form that are very focused on delivering high quality cells, and a lot of the experience out of the cancer arena has informed our ability to get great cells for trials in Parkinson's disease and other neurodegenerative disorders where this might have a benefit.
Thank you very much. And what about non-dopaminergic targeting?
So that's, again, an area where we see a lot of potential. I think the dopamine system is very well managed in Parkinson's disease, but dopamine is typically a helper neurotransmitter. It's not a direct signaling pathway. It's an enhancement to other signaling pathways. So there certainly could be other ways that we could target improving neural signaling other than just focusing on dopamine. Also, in challenges with conditions like psychosis, we typically will see, because psychosis is often a result of high dopamine levels, we don't want to use a dopamine blocking medication for Parkinson's disease psychosis. We want to look at other mechanisms. And so we've seen success with a 5-HT2A antagonist in Parkinson's. We're looking at other ways to treat those conditions.
And with discussion around some fairly complex imaging studies and lab-evaluated biomarkers today, we have a question: can that be handled by a central lab, and how can we get these complex assays done?
Yeah, I mean, we have, with a lot of these complex biomarkers, imaging assessments, they can be managed centrally, and it can actually help create more consistency across your trial. For biomarkers, a central lab can standardize how your samples are collected, processed, shipped, analyzed. Imaging can be handled in a similar way with a centralized, imaging vendor. That imaging vendor, can then provide standardized protocols, training, quality control, and central, reads, as Pete mentioned earlier, those videos. The key for this is planning, that complexity early on in the project or the trial. You want to choose vendors with the right expertise, make sure, the proper equipment can be provided to the sites if the sites don't have them. Your sites are capable, the central labs are capable. And then, just make sure there's clear processes in place for everything, sample handling, data transfer, et cetera. The more we can standardize these processes across the trial, the more reliable your data will be and the easier it will be able to manage. I know, Pete, you and I have talked before, but a good example of, this is C&D Life Sciences and their alpha-synuclein skin biopsy test. They're one of the vendors. They have a small skin, a kit, and they have that you can send to sites where they can take a small skin biopsy sample, send it into C&D Central Laboratory. They process it and analyze it. And, they also work with biopharma companies to apply that biomarker technology in clinical trials. So they're out there, and it is completely manageable.
Thank you very much. That is going to be all the time that we have left for today. I just want to assure the audience that if we didn't get to your question, I will be passing them along to the presenters to follow up offline. I'd like to thank Polly, Pete, and Amber for providing their expertise today. And you, the audience, we appreciate you being with us and hope to see you again soon. Have a wonderful rest of your day.